Speciation is the process where new species arise from an existing species. This is what generates the variety of life we see today. The world would be pretty boring without it. But how does it happen? This question has puzzled scientists for years, even Charles Darwin himself. We don't see new species popping up every day because speciation is a slow process and often cannot be observed on human timescales. But with the discovery of DNA and the ability to compare genomes, we now have a way of tackling this question. So speciation occurs when populations of the same species become isolated from the original population by a barrier. Barriers prevent populations from mating and exchanging genes with each other. They become reproductively isolated. Over time, isolated populations become genetically more different as they locally adapt to their environment. Eventually, they become so different that if the barrier was removed and they came back into contact, they would no longer recognize each other as the same species. Using DNA markers, I study how barriers influence the movement of genes between populations of the black-capped chickadee, a resident songbird to North America. Think back 21,000 years ago during the last ice age, when most of North America was covered in ice. During this time, chickadees were isolated in pockets around the ice, but when the ice retreated and the chickadees moved north, how did barriers influence their movement patterns and what happened when they came back into contact? So after analysing blood samples from 913 individuals from 34 populations across the range, I found that chickadees form 13 unique genetic groups. If there was no isolation, you would only expect one group. So on the map, each circle is a population and each colour represents a genetic group. We can see that in the west, populations are isolated by large mountain ranges, in the centre by unsuitable habitat, and in the east by large water bodies. Makes sense. But we also found genetically distinct populations in BC, where no obvious barriers exist. This illustrates that unnatural barriers can alter the speciation process as well, like the ones we create when we chop all the trees down to make space for farmland, for example. So by using DNA markers, we can essentially get a head start on the speciation process. Here we see that even a wide-ranging bird can be reproductively isolated by barriers, even after a major barrier has been removed. This gives us a better understanding of how chickadees, and maybe other species, interact with their environment and allows us to identify and protect vulnerable habitats and populations. So now the question is, will these isolated populations develop into a new species? Only time will tell.